I got into aging because I read Tom Johnson's paper showing that a single mutation in a single gene in an animal that has 20,000 genes extended its lifespan greatly, against any prediction that anyone would have made at that time.
Welcome to Longevity by Design, a podcast designed to give individuals access to the leading scientific information in the field of longevity. The ability to add years to your life and life to your years needs no opinion. Join us as we ask science to take the wheel. In each episode, Dr. Gil Blander joins a co-host and an industry expert in the field of longevity, shining a light and getting the answers to the key question: how can we live a longer, healthier life?
Hello, I'm Ashley Reaver, and I'm joined by Dr. Gil Blander. Welcome to Longevity by Design, How to Live a Longer, Healthier Life. We're produced by Insight Tracker, your science-based guide to optimizing your body from the inside out. Our guest today is Dr. Gordon Lithgow, who will discuss his research, focusing on understanding aging mechanisms and uncovering genes and small molecules that prolong lifespan. Dr. Lithgow is a professor and vice president for academic affairs at the Buck Institute for Research on Aging in Nevada, California, where we are today. He received his PhD in genetics from the University of Glasgow, Scotland, and briefly worked in biotechnology in Switzerland before becoming intrigued with the biology of aging. In 1991, he became a postdoctoral research fellow in the laboratory of Tom Johnson at the University of Colorado Boulder. Johnson was the first scientist to discover a mutation in a gene that increased the lifespan of the tiny nematode worm C. elegans. In 1995, Lithgow moved to England to start his own lab and became a senior lecturer in molecular gerontology at the University of Manchester. In 2001, he and Buck faculty members Simon Mevlov published a groundbreaking paper in Science which detailed the first use of a drug-like molecule to extend lifespan in a living animal. He moved his lab to the Buck Institute shortly thereafter. At the Buck, Dr. Lithgow became a chief advocate for capitalizing on the Institute's commitment to interdisciplinary research, and in 2007 he spearheaded and became the principal investigator for a $28 million grant from the federal government, which established the Interdisciplinary Research Consortium on Geroscience. He's credited with coining the term geroscience, which is now used worldwide to describe research focused on the connection between aging and chronic diseases.
Thank you very much.
And thank you, Gordon, to hosting us in a way, because we are doing it at the back. And we usually ask our guests, what made you to become a scientist? So we'd like to hear your story.
That goes way back. That's elementary school, I think.
Okay.
I don't know why. Sometimes different stories emerge in my head. One is that I was watching some James Bond movie, and the baddies are the all-powerful people, right? Until the scientists walk into the room. And there was some aura about the scientists that even the baddies couldn't control the scientists. I thought, that's for me. I mean, I grew up in an area of Scotland that was full of poverty and unemployment at the time. And certainly none of my teachers or fellow pupils knew what a biologist even was. And luckily enough, I was a bird watcher. And watching birds got me into biology. And I was very, very fortunate to then go off to university and be able to do this wonderful adventure in biology.
Cool. And why aging?
Oh, really, aging became interesting to me because it was a profound mystery. At the time I was in biotech, around about the late 1980s, you looked at biology and you thought, what's left? You know, we had these great strides in understanding development, especially at that point, through genetics, understanding how a single egg becomes, a whole animal. And the sort of principles for a lot of biology were really quite understood. We knew why a horse begat a horse and all the rest of it. And you looked around and you thought, well, what do we really know nothing about? And cognition was obvious, and so I tried to read neuroscientists. I'm not smart enough to be a neuroscientist. But aging, there was very little, actually. Lots of comparisons between young and old animals and things like this, young and old people, but no deep biology, no way to study it. And so I got into aging because I read Tom Johnson's paper showing that a single mutation in a single gene in an animal that has 20,000 genes extended its lifespan greatly, against any prediction that anyone would have made at that time. And I thought, we can study aging the way that we study anything else. We can find genes, we can find out what the genes are, we can find out what they do, we can solve aging. So it just was very compelling at that point that very few people were doing it, and there was a way to move forward.
From birds to now having your own lab at
Ye-
the Buck Institute,
Yeah,
which you just shared. You've been here for 20 plus years.
yeah.
So what's your research focusing on here?
Well, we like to follow our curiosity, to be honest, and part of that is doing research where you don't necessarily come in with an idea of what aging is. We let the animals we work on guide us, and we work on this. The workhorse of aging is this tiny little nematode worm, but just going to an animal and saying, what makes you live longer? And then working out why. So this has been the lab. We find mutations in genes that extend lifespan. We find drug-like molecules that extend lifespan. And then we try and work out what on earth is going on here. And that unbiased approach, I think, is great because it teaches you new biology about aging, but it also gives you potential drug candidates. It gives you a way to think about drug development by going in an unbiased way. So focus is something that we're sort of dragged kicking and screaming towards a focus. But if we have a focus, it's really around protein homeostasis and the way that protein shape changes with age and the relationship of that process, which is quite mysterious as well. Proteins are becoming damaged during aging and accumulating in different ways. And it's obviously related to disease. And that's the critical thing of how does normal aging relate to human disease.
So can you explain for our audience why working on worms is a good model for aging?
I think I can. The worm lives 20 days. And during that 20 days, it goes through just about all the biology that we go through in basic terms. So we share many, many genes with the worm. 2/3 of human disease genes are found in the worm. And we know now that the basic biology of aging, the processes that have been discovered in the worm over the last 20 years, lots of different labs, are conserved in mammals. We know that many of these processes are also driving aging processes or age-related disease in people. So that's incredible. So you can go back to the worm and say, all right, what's next? What do we not know about? And the worm is a great place to start because in 20 days, you get answers. Think about a mouse. Think about how expensive it is to have 10,000 mice. I can show you 10,000 worms today if you want to go see. 10,000 mice is an unbelievably expensive undertaking, and it's a 3 to 4 year experiment. I don't have the patience. I don't think the field has the patience. We want to be developing interventions in aging as soon as possible, and I think the worm is a great place. The success record of translating from the worm into mammals and potentially into people is very high in my opinion. That's the big hope. Coming back to why I do aging, I'm simply still curious as to what it is. And so if we are able to intervene, fantastic. If it teaches us new biology, fantastic.
And I saw in researching a lot of your work that you've made a shift from focusing on what expands life to really focusing on the specific chronic diseases that actually decrease lifespan.
Yeah, and I think that was the sort of geroscience idea. And it came conversations in rooms like this one and downstairs in seminar rooms where we were saying half of the people here are studying normal aging and they're finding it really easy to change the rate of aging. Really easy. You know, a high school student come in over the summer and find a drug-like molecule that extends lifespan. The other half of the people in the building were studying Alzheimer's and cancer and Parkinson's disease and stroke and all these diseases of late life that shorten your life. And we actually had these incredible conversations where people were saying, hold on, I'm working on that. You're working on TOR signaling? Why are you working on TOR signaling? We work on TOR signaling in flies to study lifespan. You're studying cancer. And that was going on time and time again around the building where the same processes were being studied for aging and disease. And so you take this idea that, well, maybe aging causes disease, which is sort of a geroscience idea. Aging causes disease, but it's trivial to change the rates of aging in animals. You have to put those two things together and say, does that mean it's possible to change the rates of chronic disease in mammals? And that's the grand hypothesis that I think we're all testing right now.
And what specific diseases are you looking at?
Well, my collaborations are mainly with neuroscientists, in particular Julie Anderson here at the Buck Institute, who studies Alzheimer's and Parkinson's. And that connection really came from the work that we were doing on the ways in which proteins become damaged and altered with age during normal aging. So all of us, thousands of proteins, and these proteins are behaving biochemically like proteins that we know cause, terrible chronic diseases like Alzheimer's and tau, or at least are associated with those diseases. but they're behaving biochemically like hundreds and thousands of proteins in our brains anyway. And so that was a coming together of a normal aging process of, if there's any way to preserve protein shape during aging, you see lifespan extension in worms and turns out in mice as well. And then coming at it the other way, well, these diseases are messed up in their ability to maintain protein conformation or to get rid of damage caused by aberrant proteins or toxic proteins. And so those two ideas came together. And so it's really interdisciplinary. It takes people with very different backgrounds in biology and in different chronic diseases coming together and realizing that there's a common path here. And the path is target aging. And you've got a chance of influencing the onset or the progression of chronic disease.
And the fact that the worms are more, it sounds like, from what I know, that they have their nervous system is much similar to us than, let's say, the effect of cancer and all. That's why you decided to go for neurodegenerative diseases?
I think that's fair enough. So the worm, the adult worm at least, has no cell division other than what's going on in the gonad and the reproductive system. So a bit like the neurons in our brain or muscle cells, the lack of cell division is maybe special compared to dividing cells in mechanisms of aging. And so it's natural to say that a worm is maybe a better model for a brain than it is for a tumor, for example. Follow our curiosity. Let the worm tell us what's important. Then go back to mammals and say, boy, actually, this is maybe affecting cancer as well.
Yeah.
The prediction is that there's common aging mechanisms at the bottom of all of this.
Yeah.
And, we can study DNA damage and we can study processes that are specific or we think might be specific to individual diseases. That's great. That's fantastic. But if those processes are part of the central heart of aging itself, it's likely to affect many, many more comorbidities.
Sure. I'm going to go a little bit deeper just for some biology background. Mm-hmm. When you say protein shape, just for people that don't have that biology background, what are you specifically talking about?
We take in our food and we break it down into amino acids and then we build our own human proteins. And those are strings of amino acids. And they are completely functionless unless they fold up into some sort of 3D conformation. I'll pretend this is a protein for a minute. And biochemists are great at looking at conformations of proteins and structures and making some guess about what they do. Is it an enzyme? Is it structural? And so on. During aging, what we see is that we see that proteins lose their conformation and lose their function. Now, is it important? We don't really know. We think it is, but because if we have either genetic mutations or drug-like molecules that maintain the shape, either remove the damaged protein or maintain the shape of the healthy protein to begin with, you see lifespan extension in model organisms. And again, that is a way to think about neurological disease where there appears to be the accumulation of altered protein states as well.
Sure.
And you, when we done the research, you've shown that a mild heat increasing the temperature of the worm also increase the lifespan. So that could be because of the shape of the protein?
That's right, yeah. So this amazing thing called hormesis, and the long history of this idea that if a small amount of stress, which doesn't kill you, can be beneficial. Actually, I think my grandmother used to say that. And what we think is going on here is that the stress, including a heat stress, a thermal stress, is inducing the kind of endogenous repair systems that are required to deal with the damage. So with heat, think about a fried egg. A fried egg is the denaturation of protein. It's the unfolding of protein coming out of solution. We think exactly the same is going on in cells. And that's the heat that's causing it in a fried egg. So you heat an animal a little bit, you don't get to the fried egg state, but you get to a state where you turn on the repair systems. And you have specialized proteins like molecular chaperones that bind to these damaged proteins and either refold them into their normal shape or get rid of them. So mild stress seems to be beneficial by that kind of mechanism.
And that might be why hot yoga is so popular, and it might be really good for your longevity.
It might be, I never really speculate on what's good for humans or not, being a worm doctor rather than a human doctor. But yeah, I mean, there's a great literature in things like saunas and potential health benefits and so on. And, we should always make the distinction or at least look for the crossover points between science and medicine and I guess, social activities and environmental exposures and things. I think the science behind these kind of environmental exposures is ready to really explode. really understand what it means to, as a human, to be subject to some sort of heat stress. Is it the same stuff that's going on in worms? I'll bet it is. I'll bet you it is.
And that was some of your research that you conducted in Tom Ford's lab, right?
I walked into Tom Johnson's office.
Tom Johnson, excuse me.
And said that because we were working on mutants at the time and they turned out to be resistant to heat, and that got us thinking about chaperones. And so we did these mild heat experiments. I walked into Tom's office and said, look at this, you won't believe this. Stress increases lifespan. And he sighs and opens his filing cabinet. He brings out a folder of, remember, papers were actually made from paper. And
I do.
he pulled out these papers, and it was papers on hormesis. And it was the observation that Hiroshima and Nagasaki, the exposure to radiation, of course, was absolutely terrible for almost everyone. But there was a circle at a certain range away from the initial point of the explosion where it seemed like there may be some health benefits.
Mm-hmm.
Hormesis. So there was a mild radiation of people at a certain zone that may have resulted in health benefits. I don't know if that stood up over time. It's, there's been interest in it, but that was this idea of a little bit of stress could actually have health benefits. And that was amazing for him to go back and pull out that example based on what we were doing in worms.
Yeah, and nobody should go to Chernobyl or something like that and try it.
That's
it's
correct.
still in research, but it's very interesting idea. I never heard about it. So
That's right. That gets into my category of not giving health advice. It's likely to be very bad.
So more recently, you got famous on doing some experiment and finding a few molecules that extend the lifespan, alpha-ketoglutarate, vitamin D, and iron. That basically, can you elaborate a bit about that and tell us?
Certainly, and it comes from this unbiased approach. What if we screen 10,000 chemical structures, natural products are a great place to start. They could be synthetic, they could be drug-like molecules that are already in the market, for example. Screen them and see what extends lifespan. And for me, that's always the start point. I mean, if you're not making an animal live longer, you might not be working on aging. And so we start with that principle of make an animal live longer and we'll sort it out. And that's where alpha-ketoglutarate and both that and vitamin D came out of these screens. And vitamin D, of course, we thought, well, everyone works on vitamin D. We're not going to learn anything about vitamin D. It turned out we did. We found a new mechanism of protection. And it protects proteins from losing their conformational shape. So that was great to know and could be applied to humans and the potential protective effects. And you know how complicated vitamin D is. And you know that there's detrimental effects to taking large doses. So very, very careful. Have conversations with your physician about that. But a lack of vitamin D in humans is elevating risk for a whole bunch of age-related diseases. So that was interesting. Alpha-ketoglutarate, it was the same. It came again and again and again. It was extending worm lifespan. And we cobbled together resources with Brian Kennedy here, and we set up a collaboration to look at it in mice. And this, we went into mice late on in life, which is great because that's where perhaps we're thinking about intervening as well in humans. And we saw lifespan increase in mice, and that was really encouraging in 2 different cohorts. But big thing that came out of this, and I don't understand it, and I hope another of your guests will be able to explain this to me. When we supplement a diet with this alpha-ketoglutarate, which is a natural metabolite, we have it in our cells all the time, but it goes down with age. When we supplement the diet with it, the mice are healthier. Okay, that's great. They live longer, but the period at the end of life where they're sick is compressed.
So health span is better.
Health span is better, but it's more than that. It's compression. You can imagine health span being longer, and then the period that you're sick is longer as well. Like you just stretch out the whole life course. That's not great.
No.
Think about healthcare costs. But this is an example in a mammal with that intervention where you compress the period of morbidity or ill health towards the end of life. I'm blown away by that. I never expected that. And it's really encouraging. So it's just a great example. Comes from worms in the first place, and you end up going with something that you can actually talk to your clinical colleagues and so on, and think about clinical trials in people and say, here's something that we know is pretty safe. I mean, it's a natural metabolite, and it's having this amazing effect in late life. What can we actually do in humans?
And we interviewed Brian Kennedy a couple of years ago, and he was really bullish about alpha-ketoglutarate. I think that also, if I recall correctly, in the ITP, they tested it, and that's one of the compounds that also increased lifespan there, correct?
Certainly in the CI, so there's the ITP, the mouse ITP. I can't remember the result, to be honest. That's terrible.
That's what I remember, that it's one of the only compounds.
I can't remember
Yeah.
that. But certainly when we tested it amongst different labs in the CITP, which is a worm-based, it's a model very similar to the ITP. It's a superstar. It worked in every lab, and it worked on different strains of the C. elegans, the main species that we use.
And can you explain to our audience what is the role of alpha-ketoglutarate in the body?
I wish I could. I mean, the thing about alpha-ketoglutarate is a cofactor for many enzymes, and so it's involved in all sorts of different processes, epigenetic processes, and so on. The lead author of that paper, Azar Shamirez, showed that there was a depression of inflammatory factors that could be causal for aging. That could just come along with the ride. If you extend lifespan, you're going to see inflammation reduced. So I must admit, I'm intrigued that it could actually be acting the way it acts in our high school textbooks. It's the Krebs cycle. It's that TCA cycle where it's cycling around ATP production, is producing chains of structures for the synthesis of a myriad of different biomolecules, amino acids, and so on. So I'm interested in that going back to the basics about AKG. What does it do there?
And for our audience, the Krebs cycle, and Gordon, correct me if I'm wrong, is the cycle that it's an anaerobic part that create ATP, correct, before going into the mitochondria.
No, that's glycolysis. So Krebs cycle is where you're further down, where you're after glycolysis. Then it's a cycle.
Okay.
So it's fed from different directions. It's fed from aerobic and anaerobic. But there's a big focus on aging, quite rightly, on oxidative phosphorylation, because that was the site of radical production, and the history of that idea. But actually looking at the rest of central metabolism, I think, is really interesting. And we're certainly not the only ones doing it, but I think there's a lot to be learned there. I'll tell you this, and this is essentially a collection of unpublished observations, and that is that we are finding many, many, many of the compounds that extend lifespan in worms, for example, and even taking through to really nice effects in human cell culture. They're targeting mitochondrial features. The direct targets are mitochondrial proteins. And we've got to somehow synthesize all that together into some sort of, maybe it's a new theory, maybe it's a new idea of the biochemistry of aging. Yeah. We haven't had one of those for a while.
And alpha-ketoglutarate in your research is given exogenously.
It is, it's fed in the food. This is a diet that's rich in AKG. And we don't know what cells it gets into and how much gets in there, but all those are great questions to start thinking.
Are there any companies that are trying to take it to clinical trials, or?
Yes, there is. As far as I know, there is one company that's doing formulations with AKG. Now, remember, AKG is used by people who like to build muscle, and you can buy it about 200 yards from here in a health food store. So AKG's out there.
Lots of supplements.
Lots of supplements. And if you're lucky, what's in the bottle is actually what it says on the
Yeah.
label.
Yeah.
If you're also lucky that it's formulated in a way that could be beneficial. But these are great questions, right? We had something that's just maybe is used by bodybuilders. And now we're saying, well, we know it slows aging, and we know it compresses morbidity in a mammal. Then why not think about what we can do with it now?
And if we can go for a second back to vitamin D,
Yeah.
So you showed it in worms, correct? Anyone tried to reproduce it in mice, or it was not done yet?
There's very few studies on aging, and this is one thing that we find quite a lot. If something like vitamin D, where there's thousands and thousands of publications, and you say, how on earth? What's new about what we're doing here? Well, it comes back to that lifespan question. What is it about aging that's affected here? And I think that takes you into different biology, as happened with us in the worm. Now, in mouse studies, these are crying out to be done. And I think you're going to talk to Julie Anderson here at the Buck, who's also my spouse. So we talk a lot about this stuff. And she may share with you some unpublished data on vitamin D in Alzheimer's mice. And spoiler, it's not all good.
How about iron, dietary iron?
Iron's very interesting, and Julie's done a lot of work on iron as a risk factor for disease. So I grew up not far from a steelworks in Motherwell in Scotland and played rugby. And when I was shoved into the mud and my face was shoved into the mud, the stench of iron was incredible. So I was probably exposed to a lot of iron as a kid. My father was diagnosed with Parkinson's disease, and Julie's work really showed that in mice it's a real risk factor. So how do you manage it? Well, you can manage iron. I mean, there are molecules that specifically bind iron. In worms, many years after being shoved into the mud, we showed that iron had terrible effect on protein shape. And so you could take a young worm and turn it into an old worm just by exposing it to iron. The proteins just fell out of solution. It was a very dramatic effect. If you went in with a simple iron chelator molecule, you could prevent that from happening, and you extended lifespan. So iron itself, at low doses, probably it's very essential. I mean, it's essential, right, for all sorts of enzymes. So we've got to be, again, very careful. You don't go in and take massive amounts of chelation compounds. That's an absolute disaster. But understanding more about how iron and other metals, I think it's a wide open area for research. Because if we could very specifically modulate them in specific tissues at specific times, we could maybe prevent something like Parkinson's.
Yeah, and we see it in humans. We discussed before the recording. We have more than hundreds of thousands of people, and we see that the iron metabolism in human is very complex, and we see that a lot of human, especially young women, have too low iron.
Yes.
Some people have too high iron,
Yes.
and it's not easy to maintain it. And we are looking at around 10 blood biomarkers just to understand what's going on with iron.
And something that I think you and others are finding out is that the trajectory of aging is not only dependent on so many different factors, but difficult to predict for individuals.
Yeah.
And that's going to be the trick, right? Is to say, well, you're deficient in iron based on normal clinical numbers and expert panel analysis and so on. But actually for you, that might be a good thing because your family is at risk of certain diseases associated with metals, So yeah, I really, I'd love to have a picture into the future in all these studies and actually have the mortality data. Well, hopefully no one will die. You'll be so successful that you won't ever have mortality data. But if you could, that's going to be interesting to me because coming back to the worms and lifespan, again, I think if you're making animals live longer, you really know you're working on aging.
Yeah, no, I agree.
And in your research, it's increasing the amount of iron to make sure they're not whatever the equivalent of deficient is, or just keeping them out of very high levels?
Mainly, we were studying the effect of high levels and using that as a tool to study protein conformation, and was just shocked about what high iron levels could do. I think around about this time we were feeding our infant son baby formula stuffed with iron. So that may give us pause for thought at that point.
And we do eat, especially men in the United States, eat a lot of iron from red meat.
Yeah.
Overwhelming amount. And we do see high iron is correlated with maybe insulin resistance, which many people don't necessarily want to know. But
yeah, and I think, Julie's, I think there's this new frontier that I'm sure everyone's talking about, the exposome. And the idea that we need to think about human aging, especially in terms of environmental exposures to things. We know that the genetic component for life expectancy in humans is pretty small. It's probably less than 10% for many populations, not all. So what are we doing? We need to study the 90%. And it's a very difficult problem. And, I think, again, we need to go back to doing fundamental biology in model organisms and cells to really then make predictions about how these factors are actually influencing aging in humans. Super exciting though, super important.
Yeah, so it's basically the nutrition, the environment, and the behavior.
Yeah, and possibly what your parents were up to as well. What were they exposed to that changed their epigenetic profile that's influencing us? We know that when we feed worms drugs that increase or decrease stress tolerance, we can see the effect multiple generations after the drug's gone away. So it's maybe not just us, it's our parents as well.
So do you still feel that there are more genes or protein to be identified that influence aging, or you feel like we almost saturated the field?
I mean, in one way we've saturated the field, and that is that we've got so many mechanisms now to study that so many young scientists can go around and say, I want to work on DNA damage or protein homeostasis, and that's great. And all of those studies are vitally important. But I think we haven't scratched the surface.
Mm-hmm.
I'll be honest. I mean, if you think about genes, over 600 genes in the worms are involved in aging. Wow. That sounds like a big number.
600 out of 20,000? Okay.
But that's in one condition, one environmental condition. Agar placed in the lab.
Yeah.
One strain of worms for the most part, which is used by all the labs.
Yeah.
Change the strain, and we've seen this, change the strain, you get completely different effects with drugs. And I'll bet you get different effects with genes as well. So now, you could say, well, it's too complex, it's too much. We don't want to have 10,000 genes involved. But in that search, around those 10,000, we're going to find key things. And I'm very optimistic because every time we go back, and I'm talking about drug-like molecules at this point, every time we go back and look for more, we find things that are better than the ones we had previously. And one thing I do think in looking at the activities around human aging right now is that we have quite a small list of drugs or drug-like molecules that are sort of ready for clinical trials. It's quite small.
Yeah, maybe 5.
Maybe 5.
Yeah.
If you listen to me near Barsley, there's a list with some question marks that go down to maybe 10. We need 100. Yeah. We need 200. And the ones that we've got may not be the best. If you think about how they've floated to the surface, it's great, and all this research is fantastic, but they may not be the best, and there may be much better things out there if we go back and search.
And speaking about Nir Barzilai, we interviewed him a couple of months ago, and he raised a very good point. He said that it's also the age of the animal. So some drug might be good when you are older, but not good when you are younger. And some are, like a growth hormone is great at a certain point. Metformin, rapamycin are much better later in life because early in life they can. So there is also the timing or the age of organism, and that's make it so complex.
It's very complex, and I know this sounds like an excuse for basic research, but I think you can find principles in the basic research, worms, flies, yeast, cells, and so on, that will cut through the complexity. So again, I'm repeating myself, but longevity itself, I think, is a key factor. Find that first, then you're working on aging. Then move into these other areas of different timings, different concentrations, different tissues, and then being able to manipulate individual biological processes in individual tissues, I think, will turn out to be really important. But start with, you feed this thing and they live longer, and then work forward from there.
And then there is the complexity of additive effect between 2 or 3 or 4 protein. We interviewed like a year ago a scientist from, I think, UCLA that use SIR2 with another protein together in yeast. And he showed that the crosstalk between them, depending how they work together, either increase the lifespan or not. So it can be so complex.
Absolutely. And decrease. We found that we take compounds that are beneficial by themselves. They all extend lifespan. And you think, great, let's put them together and just blow out, a huge lifespan extension and publish a great paper in Nature. No, kills them. You get over about 3 different compounds and the lifespan goes back, not just to normal, but shortens lifespan. And so it's polypharmacy, right? You're overloading the system that deals with drug metabolism. That could be a reason. It could be that the compounds are interacting themselves or the metabolites that are produced by the compounds. So there's lots of space here to get some general principles that we can take into really sort of making better predictions about what clinical trials should look like.
And if you had to bet on the 5 or 10 compound that we have today that are going to a clinical trial, which one is your favorite, or the horse that you believe that will go to the finish line?
Oh, alpha-ketoglutarate, of course. No, I mean, no, I don't. I'm certainly not the best person to ask either because of these complexities. And I don't want to make it sound like I think the worm and the fly and the yeast are the answer to everything. Of course, they're not humans, and that increased complexity, especially in drug metabolism, is going to have profound effects. I mean, the case for metformin is compelling, but it's also quite a small effect if you go back to the animal models. And so, hopefully it's a trailblazer. Hopefully rapamycin and TOR-like molecules are all trailblazers. But I think we can probably do better if we go back and find things that are fundamentally moving the needle, to quote a phrase from my colleague Simon Mello, move the needle on aging. Not just optimizing the systems we know about and having small effects on the hallmarks of aging we know about, but really moving them all in a different direction. How do we know it's possible? Centenarians and also different species. We know there's nothing about being made of proteins and fats and nucleic acids that fundamentally gives you a lifespan. We know that those same materials can be put together in ways where you can live for hundreds and hundreds of years if you happen to be a whale or a clam or something else.
Or the turtle in Galapagos.
Yes. And, and we can have evolutionary explanations as to why that happens. We don't have biochemical molecular biology explanations. Very, very few ideas there.
Yeah. What about caloric restriction? It used to be very exciting, and now, at least in human, there are more and more evidence that the data is iffy. What is your opinion?
Yeah, no, my opinion, and I listen, I'm no expert on caloric restriction, but it seems to be, it may be the way that we see a lot of interventions is that it works well in certain contexts for some people based on timing, and it won't work for others. But I mean, and certainly that's true in animal models. You'll find genetic backgrounds where it doesn't appear to be working, and you need to maybe modulate the levels of caloric restriction to actually maybe get the effects. So it's complex, it's difficult. But then, I hear clinical colleagues say the same is true for exercise, which is everyone accepts that exercise is good for you. Everyone says exercise is beneficial for aging. It's the best anti-aging drug we've got. But there are serious scientists say, well, that's all very well, but I can't tell you that it's a good idea that you exercise. I can't tell you because you're very different and you may have different responses. And some of you actually, in exercising, can be put into a very bad clinical position. So that's worth thinking about as well, is that the individual responses to these interventions that we have are really important. And so caution. Here's one way to think about it. I think that the history of aging has been one of an unfortunate cloud of pseudoscience around a core of really good, hard science. And unfortunately, I don't think that has changed. I think there's still this cloud of pseudoscience. And because of it, I think the bar that aging interventions has to meet has to be way up there with anything else that we develop as a drug, and maybe even needs to be higher. Because aging processes are involved in every other process in our body, reproduction, growth, and so on. And so maybe we need to be super careful in how we think about these things and how we roll them out to people.
Yeah,
sure.
And I think that you raised a very good point about even exercise. What we need to think about it, in my opinion, is everyone is unique and it should be personalized. The amount of exercise, the kind of exercise. Yes. Yeah, caloric restriction might not work well for me because I might not be in the right BMI or right percentage of fat or right whatever that can benefit me. So you need to look at develop a right biomarker for each intervention and then come and say, oh, this intervention is fit to you like a laser beam, and then do that. And we are not in that a situation right now, and we are looking at a cookie cutter in a way. Okay, caloric restriction is good for everyone. I assume that it's not. Even exercise. Some people, endurance exercise is good for them, some more strength, and some maybe just walking is good enough.
Yeah, absolutely. Absolutely. No, I totally agree. I mean, I think that at the moment we should be held to the standard of the double-blind clinical trial, because that's effectively how we bring things to market through the FDA and other legislation and so on. And I think that's important that we do that. But can we design clinical trials that are different or more subtle in their design from that? Absolutely. And, it's going on for Alzheimer's and other diseases right now, and the same should be true for aging.
You said the bar for aging should be really high.
Yeah.
Do you have a comfort level maybe, or something you feel comfortable saying? What is that bar? Statistically significant doesn't necessarily mean clinically significant. So what would that look like?
I think so. Right now, the bar to bring any drug to market is the double-blind clinical trial, and I think that should be true for aging interventions as well. Now, how we actually measure the effect on aging, of course, everyone's so interested in, and there's a lot of activity in that area. But certainly in terms of bringing something that's safe and effective to most people, then we have a structure that can bring that about. Now, should we have a higher bar for aging research? Possibly. And it's to do with the fact that these interventions are likely to affect multiple systems. And I know all drugs do, but especially aging, because these processes are so integral to everything in our bodies. And also we know that aging is multi-generational as well. And so we should be thinking about, and how do we do this in animal models that we can then gain confidence in going into humans? How do we know that we're not affecting the next generation? Or, I mean, if you take an intervention in your 20s, the immediate question is, how's that going to affect your reproductive capacity? Because reproduction and aging are quite tightly linked, as you know. And so there's questions right away about an intervention that's going early. If it's a bit later, well, how's it affecting the next generation?
But I agree with you, because if you look at cancer, let's say terminal cancer, so the risk reward, it can be very aggressive, because anyway, it's unfortunate, but this person might die in 6 months. So you can go with the toughest intervention in order to acquire another month. Here, as you said, it's not only your lifespan. If you are still in the reproductive timeline, it might be the next generation or the generation after. So that's why the bar should be much higher, because we are talking here a decade or maybe centuries ahead.
Yeah.
And we need to be very careful about it. Makes a lot of sense.
I mean, in the ethics class, I think a wonderful question for people who are students are really interested in aging, and we have amazing students who come. They're incredibly knowledgeable about the biology of aging and medicine as well. And I think one challenging question is that if, based on everything you see in your research in the lab, makes you think that you've got something, an intervention, maybe a gene manipulation, a CRISPR, that you could do on a baby or an embryo that you are sure is going to add 20 years to their life, would you do it? I think it's a good question. It's a complex question.
It's a complex question, because you don't know what are the side effects later. So Gordon, what are the future direction of your lab? What are you excited about? What are you planning to research in the next in the next century? No, you won't retire.
Yeah.
We will live forever.
No, I don't worry. I'll never retire. Yeah, no, I mean that. I think that I love that especially the students still come curious about the new things. So they're not necessarily, there's two ways to think about stuff that goes on in the lab. One is that you're basically coloring in someone else's picture. So the outline has been painted for you and you come in, and that's super important. This is how we're going to actually understand the complexity we're talking about. But there are some people who want to draw the picture in the first place. And that requires taking a step back from it all and saying, there's probably more hallmarks of aging. There's probably other different ways to find these. Maybe studying a different organism, studying different species, and I love that. So I like to think that we will be taking off in directions that I cannot predict, because so far maybe we've just been tinkering around the edges, and there's bigger stuff there to find. And so that's a sort of unbiased, some people may say a thoughtless approach, but it's not. I think it's just a strategy to find new science around aging.
The science lead, as you said before.
Yeah. And unfortunately, the time is running fast, so we need to get to the last question.
Okay.
And we usually ask our guests about longevity. What would you recommend our listener to do in order to live better on?
I would love to make recommendations, but I can't, because I'm not an MD. That's medicine and not science. That this is about the individual responses to certain things. And I'm shaken, to be honest, to hear that some clinicians feel uncertain about recommending exercise to an individual to a listener without an understanding of where they are and their health and their general setup. Now, I can say, make sure your vitamin D levels are normal, talk to your doctor about that. I can say, exercise for absolutely sure, have a balanced diet, all the things that we've heard about. I can say, maybe you should think about, AKG or something like that. But I can't recommend to an individual listener that they do any of those. Yeah. That's for them and their physicians to talk about. It's a shame that that's my answer. I could say a million things about what I think is going on in aging, and we've got hundreds and hundreds of interventions in freezers right now, untested in mammals. Hundreds. And if we could mobilize that, then we might have more specific answers to that question.
I think it's a great answer. It's incredibly nuanced, and how most people get health information now, unfortunately, is not through a one-on-one conversation with their doctor, with their health history,
right?
Lots of data points on their body. It's because they watch a video of someone telling them that this is what they have to do or this worked for me. So
yeah, and
it's a good point.
I think we're all subject to, all the scientists are subject to this. And I know I exercise, I have to. It would be crazy not to. But at the same time, I actually haven't sat down with my doctor and said, is this a good idea or not? I'm sure they would say yes, but I don't know that for a fact. And they probably wouldn't know either without further analysis of my health at my age. And it's difficult to navigate this.
I think that to protect or to support the clinician, they haven't been trained to help people live better long. They've been trained to basically fix a machine that is broken. To your clinician and ask him, should I exercise or not? Most likely you say, yeah, go, and without a lot of data.
Yeah, I think that's right. I love listening to physician scientists like John Newman, who's based here at the Buck. And John comes to geriatrics with a deep understanding of the biology. I really hope that that's a trend that continues. They're very rare people right now, but if we had tens of thousands of trained who understood the biology, they might be making much smarter decisions than the things I could say, because they could holistically look at their patients and understand where they are to a certain extent, and then pick and choose the information that we're giving them about interventions. Maybe combinations would work in certain circumstances, maybe not. But there's something missing there. There's a level of our healthcare that's just not there right now. As many people said, our healthcare system is really a sick care system. As you say, it's about trying to fix individual problems as they come along. The field of aging, geroscience, is all about taking a step back from that and saying, no, it's all one thing. It's all aging. Aging is the cause of this. It's the cause of the Cancer Institute. It's the cause of the geriatric psychiatric wards. And that maybe by focusing on aging, you find something that closes those wards and allows people to never see the ravages of most adult cancers.
Yeah, it's an holistic solution, and aging is basically, most of the diseases are aging-related diseases. So aging most likely is
that is the simple idea. Delay aging, you probably delay the onset of disease or eradicate in some cases, and maybe you reduce the period that you're actually sick. That's amazing. I mean, it's unbelievable, and it's an unbelievable change in our healthcare, in society in general, in our economics. It touches everything. Now, it may be wrong, and we may not be able to do that, but boy, it's worth trying.
Yeah.
It's worth trying.
Sounds like the institute that we are, the beautiful institute that we in today, that's the goal of the institute, be here and interview you, Gordon.
It's exciting to have you here. Thank you.
Thank you so much. We'd love to follow up with you in a few years and see where are you then. Thank you so much.
Wonderful.
Thanks for listening to Longevity by Design. Please subscribe to this podcast on Apple, Spotify, or YouTube. Longevity by Design is powered by Inside Tracker, a personalized health optimization platform that helps people improve their lives by improving their bodies from the inside out, using personalized, science-backed recommendations for nutrition, supplements, and lifestyle changes. To learn more, visit insidetracker.com/podcast.